So it looks like everything is set now. So good afternoon, everyone, and thanks a lot for joining this webinar series on N-nitrosamines. My name is Ranjit, and today I will walk you through some of the recent regulations which have come up in the field of nitrosamines, and I will also share with you some insights which my team has learned over the years working with N-nitrosamine impurities. So I lead the N-nitrosamine portfolio at Taros, and I was also present in the past webinar, and now this is an update, so I will walk you through the next few slides. So to whoever who are new to this topic, so I will just briefly again touch upon what nitrosamines are. So N-nitrosamine, when we talk about N-nitrosamines, these are not some new compounds. These compounds are very well known since the late 1800s. And back in the day, these compounds were ironically called as beautiful compounds. Only later, one of the first studies in US showed the detrimental effect these compounds have on exposed workers. There was a case where liver damage was reported in some of the workers. So since then, a lot of studies went into this topic, and in 1970, these compounds were deemed as ubiquitous carcinogens. And very quickly after that, the N-nitroso functionality is part of the cohort of concern, which means these compounds are or should be treated as very highly mutagenic compounds, or at least have some potency to show mutagenicity. And when we talk about N-nitrosamines in drug products, so the story began in 2018 with valsartan or the sartan families, where we found NDMA in some of the batches, and this triggered a lot of recalls of valsartan to begin with, and then also inadvertently led to recalls of many other compounds in the similar family. So in 2018, there was a lot of anxiety and ambiguity regarding what is happening, from where these N-nitrosamine impurities come from, and how they materialize in active pharmaceutical ingredients. So to answer this, the German authorities requested Dr. Helmut Buschmann to dig deep into this. He is one of our scientific advisors on the nitrosamine topic and others. So he was one who first studied the process which was used in synthesis of valsartan, and then came to the hypothesis that NDMA formation is because of the use of DMF and sodium nitrite in the process for manufacturing valsartan. And since then, this theory has been proven several times, and now since then it is a well accepted mechanism where DMF decomposes to NDMA. So since then, there was a lot of people, a lot of bodies were looking into this, and a lot of concern was poured in from the regulatory bodies and by the general public regarding the safety of drug products or active pharmaceutical ingredients. And if we just take a look at the guidelines published by EMA itself, so we are at the 21st revision. So a lot of background work has been done in this field since 2018. So a lot of studies have led to developing the regulatory framework which we currently use. And just to briefly touch upon that, as I mentioned, the story started in 2018, where NDMA and some other small nitrosamines were detected in the sartan medications. So the first phase from 2018 to, let's say, 2020 was about general awareness, what kind of compounds have these contaminants and how to deal with them. There were also some regulations published by FDA on how to deal with these impurities to begin with. So the early part of the N-nitrosamine story was about what kind of impurities are forming and how to deal with them, and what kind of regulatory frameworks should be established. So since 2020 and 2023, bulk of the work happened, and this phase we can call it like intelligence gathering and regulatory establishment. So this is the phase where bulk of the regulatory establishment happened, and we are using this framework moving on. And in the last phase, now since 2023 2024, we are talking about acceptable intakes, how to assign acceptable intake values, and so on, and how the risk mitigation and compliance aspects of these N-nitrosamine impurities are. So this is a very short and brief summary of how the evolution of regulatory background has happened in N-nitrosamines. So When we talk about N-nitrosamines, we should understand there are two different classes of N-nitrosamine. One is the small nitrosamines, which was very well known in sartans and so on, and on the other hand of we also have API derived N-nitrosamines, so which are called as NDSRIs. So when we talk about small nitrosamines, a lot of animal carcinogenicity data is available, and a lot of studies have gone through in understanding how these molecules interact with the biological system and their mutagenic potencies and so on. But when we talk about NDSRIs, very little or no data is available. So now it becomes very critical to know what kind of acceptable intake limits you need to begin with. And then we also need studies in parallel to understand what kind of mutagenic potency each compound can exhibit. So if we take a look in the figure, if we consider rasagiline and phenylephrine, they are equally potent as the small nitrosamines, and N-nitroso, lidocaine, and beta histine is also equally, if not more potent than some of the small nitrosamines. But there is a huge window. If you take a look at imatinib, benizapril, they are not as potent, or they are not classified as potent as some of the other NDSRIs. So again, there is a ambiguity in knowing or assigning the acceptable intake values, and so this is something which is now the cornerstone of the regulatory background. So before we go into that, we can just take a look at how the metabolic activation to potency or mutagenic defect actually work in nitrosamine. So in any N-nitrosamine is not a toxic compound or a potent compound by itself. So upon interacting with the biological system, more specifically with the liver enzyme P40, P4, P450, the N-nitrosamine is hydroxylated to give an alpha hydroxyl species. This then quickly loses an aldehyde molecule to give disonium hydroxide, and this disonium hydroxide then dehydrates to give a disonium ion. And now this disonium interacts with DNA, and then this is the one which is responsible for the genetic defect or genetic mutation, which can then lead to carcinogenic effect. So the alpha hydroxylation step is very crucial when in terms of determining the potency. And since it's a chemical process, of course, it is dependent a lot on electronic and steric factors. And now this is where we take this understanding of how the mutagenic potency is activated by nitrosamine, and this is what you use to assign acceptable intake of certain NDSRIs. So, as I mentioned, I'm not going to go deep into it. So, there are several factors through which the molecule can be considered to be more potent and less potent. For example, presence of alpha hydrogen. So, the more facile the alpha hydroxylation process, the more facile or the more potent is the nitrosamine. That's a general understanding. And there are several other groups such as carboxylic acid, pyrrolidine group, which decreases the potency because then it doesn't make the molecule as susceptible to alpha hydroxylation mechanism than a typical nitrosamines, such as some of them are like this. So if you want to go deep into this, Emma has published this SEPA guidelines last year, and it's a very briefly and very nicely described in their dockets. And I would encourage, if you are interested in understanding how these molecules are classified, then this is one good source where you can always refer to. So, since now we are talking about drug safety, so in general, everyone's safety, so there are certain responsibilities which have been set by the health authorities for the market authorization holders and the API pharmaceuticals, and this is also heavily described in the EMA guidelines. But we can go through. So, first and foremost, you have to do a risk assessment, testing confirmation, regulatory reporting. So basically, identification, then analytical testings, validating the analytical methods, and so on. And at the end of the day, you have to comply or adhere to the regulatory backgrounds which the responsible health authority you are dealing with has set. So to understand this a little bit easy, there is a three-step in general. It's a general accepted mechanism where you go with a three-step evaluation for risk assessment. So in the first step, you do risk evaluation based on what kind of process is used, what kind of degradation can happen, what kind of solvent sources are used, what kind of cross contamination can happen, what kind of formulation is done, what kind of excipients are used, and so on, so forth, and so on. So if you say that, okay, I have a secondary amine, I have a nitrite source, and I have the right conditions to trigger the nitration pathways, then you go to the second step. So second step is where bulk of the work which I do in my lab happens. So we are responsible or asked to identify and characterize the nitrosamine impurities in certain molecules. To be more precise, we study nitration pathways, what kind of products we get, and we were very surprised over the years that we have seen a lot of deviation from the typically accepted nitration pathways, which I will share in the later slides. So then, of course, in the second step, then you confirm, you validate analytical method, then you also see for small nitrosamines and NDSRIs. So now when we talk about nitrosamines, it consists of both small nitrosamine as well as the API-derived nitrosamines. So then if you have done all the due diligence, then you move to step 3, where we talk about mitigation strategies, and then you can also argue about skipping the testing or omitting the test. So there are some general principles which work across all the health authorities. So if your N-nitrosamine contamination is more than 30% of the acceptable intake. Now, the acceptable intake value comes from the SEPA guidelines, which we will discuss in the next slides. Then you have to specify what kind of nitrosamine is there. Then you have to develop all the methods, then validation, and then mitigation strategies have to be communicated. If your contamination is less than 30% of the AI, then you can ask for skipping testing. But if your contamination is less than 10% of the acceptable intakes, then you can completely omit testing. And then there are some nuances to this, so don't take this as a blueprint for determining what kind of needs you have to fulfill. But the overall general framework is something like this. So then you again report the changes to the authorities, and then you can take the ball rolling, and then eventually you have to ensure that your drug is free from N-nitrosamine, or at least the contamination is not above a certain limit. So now this is something very important. So I found this figure on the website for EMA, and this very nicely summarizes how to deal with a new nitrosamine. Let's say you're developing a new product, and you see that, yes, I have a new nitrosamine which forms. So now the first question you ask is, do you have sufficient animal carcinogenicity data? If the answer to this is yes, then you go through TD50 value, and then from the TD50 you can assign an acceptable intake. Which is very rare. In many cases, a lot of data is not available. So if your answer is no, there are several options on assigning the acceptable intake, and based on the acceptable intake, you can then determine what kind of analytical testing you need to do. So the first option is this SEPA approach, where you assign the potency using the SEPA guidelines. There are 4 different categories or 5 different categories. So the most potent is 18, has an acceptable intake of around 18 to 26 nanogram, depending upon which health authority you follow. And the least potent is around 1500 nanograms per day. So if you don't have any data, so this is a very good guideline to assign what kind of potency category a compound falls into, and then you can decide the further intervention points what you have to do. If not, then you can also do a GLP compliant Ames test. If the Ames test is positive, then again you have to go to the SEPA guidelines to assign the values. If they are negative, then it is by default going to 1500 nanograms per day, which you can see on the right side of the screen. If you don't, another option is to ask if you have a surrogate nitrosamine available. If the surrogate nitrosamine has enough data, then you can assign the acceptable intakes by SAR and the read across parameters. If not, there is one more. You can also always go for in vivo mutagenic studies. And again, there are several options where you can, depending on what kind of data you get and depending on what kind of health authorities you are engaged with, you have a certain degree of further regulations which you have to follow. But this is a very nice summary on how to deal with, let's say, new nitrosamines. And based on this SEPA guidelines, we have an acceptable intake value. Based on the acceptable intake value, you can determine if you have to do testing, if you don't have to do testing, and so on. So now it's a very solid framework which works very well. Now, when we talk about N-nitrosamine, so the sources of contamination, if you want to list each and everything, it's a huge list from where the N-nitrosamine contamination can happen. So, but just to briefly summarize, I would say there are three categories. One is packaging material storage conditions. These are probably the least offenders or the least probable sources of N-nitrosamine, because we understand a little bit more about how the storage condition packaging material can affect. The second one is manufacturing process and recycled solvents. So these are the major sources where you can contaminate your compound during the manufacturing process. And if you use recycled solvents, you use water which is contaminated and so on. So this can be one way of increasing the contamination. But one of the most debated, or the one of the least studied, are how these the excipients and drug substance compounds. So these are heavily debated because a lot of excipients have certain nitrites, and there are not very strict regulation when it comes to excipients, and there is a little less understanding on how the mechanistic pathways work when the amine in question interacts with excipients and drug substances. So in many cases, formulation chemicals can also have an effect. So that the nitrosamine comes at a very late stage of your in your project rather than early stage. So you have taken all the proper steps, and by the end, because of the excipients and lack of awareness on about the nitrite contamination and nitration pathways, this can lead to a lot of uh, the mechanisms for nitration are not very well known. So there's still a lot of debate going on in this topic. So to address this, very recently, IPEC and Lhasa have summarized some of their findings. So the key points discussed were about mitigation strategies, how this can be done. It's a very nice document. You can I also encourage the viewers to go through once. Now, the problem here is the suppliers should provide enough information to the MAH bodies or the pharmaceutical producers, but still the testing for nitrosamine is not required when they are supplying certain raw materials. So to address this, now Lhasa has a very detailed database where you can get a fair idea of what kind of nitrite contaminations are available or possible in your excipients, and what kind of excipients to choose from. And in this meeting, a lot of big companies came together and voiced their opinion regarding what should be done. So one BASF said supplier selection is very critical, and there is still need of optimizing the ion chromatography. The ICMS methods are not yet up to the mark. So a lot of things are desired when it comes to understanding how the excipient and the formulation affect your active pharmaceutical ingredients, or can cause the nitrosamine contaminations. And this is where laboratories like us help a lot. We study the mechanisms for nitration, and we also do thorough characterization and analysis of the compounds to add to the understanding, and which then helps in it's not just our lab. There are many labs around the world which provide data, and then these data can form the background for a regulatory update. And since we are working, I think probably for the last 4 years with N-nitrosamine, so we have developed a certain kind of understanding on how the nitration pathways work. So let's take an example about risk assessment and susceptibility studies. These are the studies which we usually carry. So the first question we ask is, do we have a secondary amine to make the nitroso-ciprofloxacin? Yes, the answer is yes. Do we have the nitrosating conditions in acidic medium? Yes. Then if the answer to both of them is yes, and if everything works well, then you synthesize, then isolate and characterize the N-nitrosamine of the NDSRIs. So these are the cases where everything works fine, and but the another problem which we have seen over the years is uh, this nitrite induced degradation or acid induced degradation pathways are some things which are never discussed or never studied thoroughly to understand what kind of nitrosamine contamination it can add to. So let's take an example where we had a compound where we had a secondary amine and a tertiary amine. And when we did our reactivity assessment and force degradation studies, instead of seeing the desired nitrosamine, along with this, we also saw a lot of other nitrosamines which were forming. Now, of course, the selectivities are different. The degradation extent of degradation study depends on how long you run the program, what kind of conditions, what kind of forced conditions you are putting in and using. But if you take a look at all of these nitrosamines, they fall in a different potency category. Now, it is very critical to understand what kind of N-nitrosamine we are dealing with, and if you know very well what kind of N-nitrosamine you are dealing with, then you know the potency category, and from the potency category you can know the acceptable intake, and from the acceptable intake you then decide what your mitigation strategies will be. And so it is very vital in understanding these nitrite induced degradation and other deviations in this mechanism. So another critical part of the N-nitrosamine topic is analytical capability. So let us just, for example, if you take a look at, if your maximum daily dose of a medicine is, let's say, more than 1 gram, and you have one of the most potent nitrosamine impurity contamination, let's say 18 to 26 nanograms per day. So your LOQs for such compounds are around 0.02 ppm. So when we are talking about or dealing with such small amounts of nitrosamine contamination or nitrosamine detection, you need a very strong analytical support. And in that sense, we are very lucky to have a very good analytical and strong analytical team, which are constantly trying to provide solutions. And very recently, we have developed some certain methods where we can easily distinguish between amino sugar nitrosamines, which was in the past very difficult to understand. And based on this, we can now also isolate some of these components, because we understand how the mechanism work, how the analytical methods to be developed, and then we can also work on with uh, isolation and further characterization, because for characterization of these molecules is again a big challenge, and when it comes to understanding the structures. Then I will now share again some of the case studies where we have seen deviation in nitrosation mechanism. So we also had another compound where you had a secondary amine, then we had facile nitrosating condition, and we were expecting to get N-nitrosamine, which is run-of-the-mill secondary amine, is nitrosated. However, in this particular case, we observed that instead of the N-nitrosamine impurities, we are getting a cyclized product. Now, when we have a cyclized product instead of N-nitrosamine, now we are talking about some impurity which is not N-nitrosamine. So you don't have to go through the trouble of actually going through the or designing mitigation strategies and so on. But if you take a look at the molecular weight of both the compounds, they are identical. So it is very easy to misjudge N-nitrosamine impurities with something else which is very close and which is nitrite induced, So finding the right structure elucidation is, I believe, is very critical, because it can make or break a risk assessment of your N-nitrosamine compounds. In another case study where we were trying to nitrosate a secondary amine in presence of primary amine and an amide. So here we observed that instead of getting the desired compound, which is the secondary amine nitrosated, we found a hydroxyl N-nitroso compound. Now, when we talk about the hydroxyl moiety is if we go back to the SEPA guidelines, hydroxyl group decreases potency. So if you if you take a closer look, the potency categories of both these compounds are different. So if you assume that you will be getting the certain type of N-nitrosamine which has a certain potency category X, but now you are talking about a potency category Y which is less than X. So then again, this will have a significant impact on the project timelines, and you don't need to go through such heavy or extensive mitigation studies to support your findings. In another study where we observed that certain N-nitroso species can be reverted under certain conditions, or can be influenced to revert back. And this is a very new study, and this is something which can help a lot in understanding what kind of, instead of going through the whole process of risk assessment, we can also understand or try to get a fair outlook in developing such strategies where you can degrade your N-nitrosamine impurity selectively to get back the amines. So there is still a lot open in this topic. We just found this result, but this is something which is very crucial in furthering the understanding of N-nitrosamine impurities and so on. In another example, again we have done a detection but failed attempt in isolating the desired N-nitrosamine. So in this case, we observed that the N-nitrosamine forms at a certain interval, and which then converts into the compound which is on the very right of your screen. So again, now we are not talking about a nitrosamine impurity, but we are talking about something totally different. Now, the key importance of these findings, which we have learned so far, is now we are talking about some impurity which is not nitrosamine, but we still don't understand what kind of genotoxic or what kind of mutagenic effect these impurities have. So this can also help in further understanding and further discovering new impurities which can either have a detrimental effect or can be totally benign. So this is still a vital step in risk assessment strategies when we talk about our actual medical products. So, just to give a rough idea of what we have been doing so far. So, we have been working with N-nitrosamine since late 2020, maybe early 2021. We are in full steam, and since then we have done targeted synthesis and long-term stability studies of around 100 plus compounds. We have investigated LODs of APIs and nitrosamine derived from APIs around 130 odd. And then overall, we have done reactivity assessment, stability test, and stability studies of 150 odd compounds. So I believe that we are one of the most experienced lab when it comes to understanding and handling N-nitrosamine impurities and APIs. So this is something which I'm really proud of, that we are able to further the understanding of N-nitrosamine impurities. So how we do it? So just to give a brief overview of how we do it. So when a new compound under question comes to us, we first do a susceptibility assessment where we study or where we monitor the nitrosation pathways using process similar and forced nitrosation studies. We also evaluate LODs of the actual starting material to understand at what degree we have to titrate our analytical testings. And once when the answer for this stage is yes, the compound is susceptible, then we go for chemical synthesis. So far, we provide from 1 to 10 gram of analytical standard. In rare occasions, we have provided 200 grams or 250 grams of N-nitrosamine impurities for Ames testing. So then we then we thoroughly do structure elucidation using NMR, UHPLC, and several orthogonal techniques. We also evaluate the LODs of N-nitrosamines, and once you confirm the LODs and the structure, then we again revisit our susceptibility assessment, and then we can confirm what kind of nitrosation pathways and what kind of nitrosamine impurity is forming or not. In addition to this, we also do solubility test and stability studies and force degradation studies, where we do thermal long-term stability studies. And for solubility test, we do organic solvents and aqueous buffers. This helps sometimes in some customers for understanding what kind of dilutions they need to use for AIMS test. In addition to this, we also have consultancy service where we propose risk mitigation strategies once you have identified the nitrosamine and there is still a limiting factor on getting rid of the nitrosamine. So this is where our consultancy services comes into play. So to now, I'm at the end of the presentation. So in working with the N-nitrosamine impurities, we have developed certain expertise, and this is just a summary of what kind of services we offer. So we established in late 2020 and have so far delivered more than 100 plus 100 plus different nitrosamines. We also have a dedicated facility in-house to deal with nitrosamine impurities. And the services which we offer range from early stage screening, synthesis and isolation, strategic consultancy, thorough risk analysis, and many times we provide documentation which our partners use directly for sharing it with the health authorities. And we also provide long-term stability studies, force degradation studies, and in-house we have a dedicated team which works with nitrosamine impurities and uh, which is very which are very which have a very keen eye in understanding and studying the nitrosation pathway. So this has led so far to all the success in the nitrosamine field. Last but not the least, we also help in supporting AIMS testing through our biological testing partners. So this is the end of my webinar. So before I go to the final slide, I would like to thank the present and past members of the N-nitrosamine topic at Taros. And if you have any questions, please feel free to connect with me on the emails given below, or you can also connect with LinkedIn Taros account, so you can also ask your questions there. So I hope you guys enjoyed the webinar, and thanks a lot again for joining. And hopefully we will have a new webinar coming, and you guys will hear about it on our website very soon. So thanks a lot.